We're so excited. This is our inaugural Ken Coit seminar series. And we have an illustrious guest today, which you'll hear much more about him when Ken introduces him. But before we get started, I just wanted to welcome everyone. I'm Bonnie LaFleur, and I would like to introduce President of the University of Arizona and the United or maybe, I don't know, he might be on his way. You heard it here first. Announcing the candidacy here. So, Dr. Garamella is going to introduce our first speaker today.
You know, I saw Bonnie as she walked in and she said, I have to introduce you and I hope it all goes well. I said, how can that go wrong? But you can see how it can go wrong. So different president, folks. Faculty member, one of you, happy person. So anyway, greetings everyone, thank you. I'm so pleased that this room is so full. This is one of the better buildings we have on campus. We've had a board meeting here. We had a bunch of posters outside and a reception in here. So Andrew Weil and his whole team, and they've done a fantastic job. They've put us on the map. They were into wellness before it was cool. And so I'm just pleased to see this particular talk and this topic being discussed here. So, you know, my introduction to rapamycin came in multiple ways, but Ken gave me a book called Outlive by Peter Attia. Some of you, if you're here, maybe you've read that book. But then Hesham Sadek pointed out another book for me. It's called Chasing My Cure by Dan Feigenbaum. How many of you know that book? Great. So look at it. It's a very easy book to read. I read it in 2 sittings. It's called Chasing My Cure. Feigenbaum is spelled with a J. He's a professor at Penn, I think. So anyway, he was afflicted with disease that he couldn't, with cancer, like 4 bouts of it, almost died 4 times, and was a football player, decided to find a cure himself, and did. And something to do with rapamycin. So anyway, I'll let you read the book. But so it's fascinating that, you know, on NPR a couple of days ago, I was hearing about people setting aside trusts for themselves for when they come back. They're preserving themselves in cryogenics and don't want to come back poor. I don't know if you heard the NPR article. It was very interesting, kind of ghoulish and weird. But anyway, so lots of interest in aging, anti-aging folks, and I think it's great that Ken and Donna Coit have been just great supporters of this university. Obviously, our pharmacy school is named for them, but their generosity has been really it's crossed boundaries, and he named our athletic director, who's named the Ken Coit athletic director, for example. So we're just so pleased that Ken and Donna are so generous to us and are so curious. I think Ken, anyone who chats for a few minutes with Ken knows he's insanely curious about any topic, whether it's fusion energy or AI or pharmacy or whatever. Apparently he never practiced or practiced for 3 years or something, but it's kind of gone on to do other things. So Matt Kaeberlein, I've heard a lot about you. We're honored that you're here to speak about this. I think one of the things that I think about such talks and such work. So this is the first clinical trial that sort of randomized clinical trial that's going to be conducted on rapamycin, and Matt will be the scientific advisor for it. I'm really pleased that it's being done here, and you know, we're all, at least some of us who are getting a little older, Rick, we feel like maybe there's something to be learned from all this sort of stuff. But it just is great. It's part of our land-grant mission to be supporting our community with lectures like this and with work like this. I think the exciting part of this is that there are multiple sort of centers and colleges and such that have come together to do this work. And I'm just so thrilled that we have work like this being done here. Thanks to the support of Ken, but there's so much more. I hope that any of you that's not from the university will continue to sort of explore all the things that happen at this great place, because behind every door there's some great work being done. I went through every college recently and spent several hours there. Went through the pharmacy school, saw your amazing museum. That was just great. If you've not seen the pharmacy museum, and if you like mortar and pestles, you should definitely go there. It's really interesting. Anyway, thank you all. Enjoy a great talk. Thanks for coming. I screwed up too. I'm supposed to introduce our Ken Coit, our next speaker, and of course the reason we're all here. So thanks, Ken. Please come on up.
Thank you, Suresh. I graduated from here 59 years ago, and I had no idea that I would ever do this sort of stuff. And making the kind of capital I have made for my clients and myself over the years has been it's pretty fantastic, and it's really great to give back. And I think this rapamycin study we've got coming is I think is like Matt Kaeberlein, who's going to be speaking next, says it's our best shot on goal in terms of longevity. And I got to bring up this article out of the Wall Street Journal on May 8th, 2025. It talks about ultra-wealthy pile money into longer lifespans. These people have put in about $5 billion into the lifespan research. And I think we're going to do better here than they are. Let's see if we're right or not. Anyhow, thank you, Suresh. It's great talking about drug research. It's near and dear to my heart. I consider myself a scientist first, a businessman second, and one of my greatest heroes in the business world is Warren Buffett. Everybody knows him. He went from 84 to 94. He made his clients billions of dollars, and he gave away hundreds of millions of dollars. I really like that formula. Now, if I can emulate that over the next 10 years, that'll be great. My poor wife, she's probably tired of hearing me saying that I'm going to retire in 5 years, but I'm not. Longevity is all about living better and living longer, and especially the better part. So what's the secret? What if I told you the answer that this tablet prompts the body with a single message? Hey, act like you're starving to death. This tablet sets off a chain reaction that causes your cells, our cells, to kick into high gear and embark on an intense cleaning spree. And from a scientific perspective, it's called autophagy. And I would definitely look it up on ChatGPT or Anthropic. This is what the secret, I believe, of rapamycin is. So what autophagy does, it recycles damaged proteins, repairs worn out mitochondria, essentially decluttering the cellular attic and cellular junk, making you more healthier. And as over time, your autophagy gets less and less as you get older. So I'm hoping that it works for me down the road. We'll see. Anyway, the power rapamycin is an mTOR inhibitor. It silences the aging process, allowing our bodies to rejuvenate. It's like hitting the snooze button on Father Time. The evidence is clear. We're not just speculating here. On every living thing that has been studied, from cells to multicells, to mammals, rice and rats, and there's a dog aging study coming. They have lived extended lifespan by astonishing 30%. Now, it's like turning an 80-year-old. I don't know who's 80 here besides me, but you get out to be 104. And so you can ask this 104-year-old, what's the secret? And have them reply, I'm taking this tablet and I avoid kale. We all know kale is not that good to eat. Imagine if we could slow down aging at the cellular level, shifting our focus from merely treating diseases and fundamentally changing the aging narrative. Think of upgrading your flip phone to a smartphone. So now let's get together to a champion who's doing groundbreaking research. It's my great pleasure to introduce Matt Kaeberlein, who has a PhD from the University of Washington. He's written a mere 250 papers on aging and longevity, and he's been studying rapamycin for about 20 years. He started the 8 Dog Aging Project in 2014. And again, like I said earlier, his best comment was he thinks this is the best shot on goal. Matt Kaeberlein.
All right, well, Ken gave my talk for me, so I'm done. So first of all, I want to thank Bonnie and especially Ken for inviting me to give this talk. It really is an honor, and coming from Seattle, I'm enjoying seeing the sunshine. So I'm going to really try to give you sort of an overview of the evidence that rapamycin slows biological aging and kind of where we're at today in terms of what we know about rapamycin in the real world and why this clinical trial, I think, is going to be immensely impactful. So just to start, what is rapamycin? So rapamycin is a small molecule. It was discovered on Easter Island, which is also called Rapa Nui. That's where the drug gets its name from. And it's actually produced by a bacterium found in the soil on Easter Island called Streptomyces hygroscopicus. It is a specific inhibitor of a protein complex called mTOR complex 1. And rapamycin seems to be extremely clean biochemically, meaning it is an inhibitor of mTOR complex 1, and as far as we know, only mTOR complex 1. mTOR actually stands for mechanistic target of rapamycin, so they weren't very inventive when they named the protein after the drug. And one of the nice things is rapamycin has been FDA approved for 27 or so years. So we know a lot about rapamycin and its impact on human biology. This is a little bit of a double-edged sword, though, because it was first approved as an organ transplant drug. And so it was been used in that context, high doses of the drug in people who are taking strong immunosuppressants who needed an organ transplant. So a lot of the information we have about rapamycin is its impact in that context, and I'll come back to this. So originally approved for kidney transplant, then approved for other types of solid organ transplants, also for cardiac stents and a few other things. I'm not going to try to pronounce the L word there. And then some derivatives of rapamycin, which are called rapalogs. And for the purposes of this talk, I think you can think of them as identical to rapamycin. Things like everolimus or temsirolimus have been approved for certain forms of cancer and genetic condition known as tuberous sclerosis complex. So lots of information in people about rapamycin. Okay, so that's rapamycin. What is mTOR, the target of rapamycin? mTOR is a kinase, so it phosphorylates. Did we lose our slides? That's okay. So it phosphorylates other proteins. It's involved in cell signaling. And I think, again, for the purposes of today's talk, maybe best to think of mTOR as sort of a gatekeeper that the cell uses to make a fundamentally important decision, and a decision that every organism that has ever lived has needed to make. And that is it a good time to have babies, right? And so from an evolutionary perspective, you can see why that's a really important decision to get right. And if you think about what do organisms need to evaluate to make that decision well, it's not a lot of things. It's how much oxygen is available if you're an oxygen-using organism, temperature suitable, and how much food is around. And mTOR really helps the cell sense all of those things, but is best known, as Ken alluded to, as a nutrient sensor. Okay, so when there's lots of nutrients around, mTOR activity gets dialed up. That's a signal to grow and reproduce. When there's not very many nutrients around, mTOR gets turned down, and that's a signal to shut down reproduction and turn on stress response pathways. And the thinking around this is that it's good to turn on stress resistance because you want to outlast the famine until food is available, and then you can enter growth and reproduction. It turns out that stress-resistant state also is a slower aging state, we think. So that's an evolutionary explanation. Ken gave a molecular explanation, autophagy. That's an evolutionary explanation for why mTOR affects aging, and rapamycin, because it's an inhibitor of mTOR, mimics that nutrient-deprived state and turns on these stress-responsive, turns up autophagy, turns down protein translation, and does a bunch of other stuff, which makes it complicated to really figure out what the detailed mechanisms are. Okay, so how did we start to think mTOR might be important for aging? I feel very fortunate to have been involved in that discovery. I wish I could say that I was smart and I thought mTOR is the answer. I actually got interested in mTOR and its role in aging because I was doing an unbiased genetic screen, and mTOR happened to come out of the screen. So I had no clue. Other people were smarter, and this was a really interesting time because 4 labs using 3 different organisms all converged on mTOR at the same time, independently. That happens in science sometimes. So 2 labs in C. elegans, 1 in fruit flies, and then the work that Brian Kennedy and I were doing in yeast, we all found that if you genetically turn down mTOR, you slow aging and increase lifespan in all of these organisms. And I think all of us were like, wow, that's really cool. This is 2 billion years of evolution. This must be important, right? If it's affecting aging across all of these organisms. Now, the people who study humans and study mice were like, you guys are working in flies and worms, and that doesn't matter. But then in 2009, so 5 years later, a study was published in mice. And I really think this is certainly one of the top 10 most important studies in the field of longevity science. So this is a study that came out of the NIA Interventions Testing Program. For those of you who aren't familiar with that program, it is a program that tests anywhere from 5 to 7 interventions, usually small molecule drugs, in mice every year at 3 different sites. So it has triplicate replication built in. Anybody in the community can nominate a drug to be tested. It goes through a selection process. I'm actually on the steering committee for the ITP, so it goes through an academic review process. The 5 to 7 are selected every year. Rapamycin was in the first group of drugs that were tested in the interventions testing program, okay? And there was a happy accident here. So they start all of the interventions at between 6 and 9 months of age, so young mice. And it turned out when they were trying to formulate the rapamycin, they couldn't figure out how to get it in the food to deliver to the mice. So they had to start the experiment because they had thousands of animals ready to go. So they started the experiment and they're like, well, we'll get it figured out in a couple of months and we'll add the rapamycin then. It took them about a year. And so by accident, these mice started getting rapamycin at 20 months of age. So that's about a 60-year-old equivalent in a mouse. So that's what's shown here. The red line are the mice that got rapamycin. The blue line are the mice that didn't get rapamycin. And that arrow is when they started getting the drug. And you can see the curves immediately diverged. So that was cool. But I say it changed the field because before that, if you'd asked me or almost anybody in the field, is a drug going to work starting in middle age? They'd be like, no, you have to start when the animals are young. So this was really the first demonstration that biological aging is malleable all the way through middle age. And now that's kind of accepted, but at the time, this was a fundamentally important observation that told us something about biological aging that we didn't expect. And what have we learned since then? So a lot, obviously. I don't have the time to tell you everything we know about rapamycin, but some of the things that are important to appreciate is, so that was 2009, so we're in 2026, so 17 years. Rapamycin is still the most effective longevity drug we have, period. It has the largest effect size, it's the most reproducible, it seems to affect the most health span metrics, at least in laboratory animals. And in some ways, that's a little bit disappointing. I would have liked to think 17 years on, we'd have something better than rapamycin. But for now, at least, it's still the most effective drug we have. As Ken alluded to, it slows aging in every organism where it's been tested. That doesn't mean it's going to slow aging in every organism, but in every organism where it's been tested, it has worked. Increased lifespan, improved health span metrics. In mice, as Ken alluded to, he gave my whole talk for me. It increases lifespan up to 30%. That's pretty good. That's pretty good. It's the best we've got for a longevity drug. Caloric restriction can do better, but in terms of drugs, that's the biggest effect we've seen. I already mentioned it works when started in middle age. It seems to impact every hallmark of aging. So for those of you who are familiar with the hallmarks of aging, it hits all of them. And that's what we would expect for something that is fundamentally modulating the biology of aging. And as I've alluded to, and as I'll show you in a minute, it appears to broadly delay aging across most, if not all, tissues and organs, and in some cases, even reverse functional declines that have already happened in an aged animal. And to me, from a translational perspective, this is what is extremely compelling. So I think that's been best demonstrated in the heart, the immune system, the ovary, the oral cavity. I should probably add brain and skin on there as well. And I'm going to show you just a little bit of data for heart, immune, and oral cavity. Okay, so this is actually work that came out of my lab. Graduate student at the time, Jonathan Ahn, who's now a professor at the University of Washington. He's a DDS PhD, so as a dentist, also got his PhD. And to be honest with you, I had never thought about oral health in the context of aging biology before John came to my lab. And he's like, well, periodontal disease is an age-related disease. It's probably related to the biology of aging. So the first thing he did was took mice and just asked, if we just let them age, so no intervention, Do mice show declines in oral health and show symptoms of periodontal disease with age? And the answer was yes. So that allowed us to do a really cool experiment and say, well, what happens if we give those mice rapamycin? And what John showed is we could reverse the clinically defining features of periodontal disease in mice, starting treatment at 20 months of age, in as little as 8 weeks of treatment. And so he looked at 3 primary features here, one being gingival inflammation, so gingivitis. That was reversed. The age-related increase in gingival inflammation was reversed by rapamycin. He looked at the change in the oral microbiome with age. We could show a reversion of the composition of the oral microbiome back towards the youthful state. And what I think is most compelling is he could actually see bone regrow around the teeth in these mice. So it's probably a little bit hard to see in this image here, but if you see those 2 yellow arrows, if you look at the top image, you can see there's a gap between the bone and the tooth. And it's the same mouse. 8 weeks later, you can see there's bone filling that gap. So this was pretty compelling evidence that rapamycin could restore oral health in an aged animal. John is currently looking at this in people. This is looking now at heart function. So 3 or 4 different labs now have shown that you can reverse age-related declines in heart function. This is from Peter Rabinovich's lab, who was at the University of Washington at the time. So this is done by echocardiogram, which is ultrasound for the heart. So we're looking at 2 different features here. And this is a little bit of a complicated graphic, so I'll walk you through it. So the design here is you have young mice or old mice. The old mice are also 20 months old, so about a 60-year-old equivalent mouse. And in the old mice, they either get a placebo, so that's the control, or rapamycin. Okay, the rapamycin is the old rapamycin group. And the measurements were taken either before that 8-week period or after that 8-week period. The white bars are before the period. The black bars are after the period. Okay, so just a couple things to note. The first one is left ventricular mass index. That's a measure of cardiac hypertrophy, which happens with age. If you look at the young versus the old, you can see those bars are higher. So there's an increase in hypertrophy with age in the control animals, the animals that never saw rapamycin. And then if you compare the white to the black, what you'll see is that in the control animals, there's no change, right, in LVMI. But if you look at the old rapamycin animals, after they got rapamycin, LVMI comes back down to where it is in the young mice. So this is a reversal of age-related hypertrophy. This has now been seen, and I'll mention this in a minute, in dogs and cats, and some evidence even in people that rapamycin is effective at reversing age-related hypertrophy. The other metric is something called E to A ratio, which is a measure of how well is that left ventricle contracting and pumping blood. And again, if you look at the young versus the old, you can see those bars are lower. So left ventricular function declines with age. And then if you look at the old rapamycin group, white to black, you can see there is a reversion back to the youthful level. So also for left ventricular function, it seems as though rapamycin is reversing age-related declines in cardiac function. And this is my favorite of the experiments I'm going to show you. I like this experiment even more than the 2 from my lab that I'm going to show you. This is work from Pan Zheng's lab looking at immune function. This is obviously, we all learned during the pandemic, immune function is really, really important, especially in the elderly, right? So their question was, how does the immune system change functionally as mice get older, and does rapamycin impact that? And again, this is a little bit of a complicated graphic, so I'll walk you through it. So what they did was they took either young mice or old mice. They either gave them an immunization for influenza or not, and then half of the old mice got rapamycin. And the question was, 2 questions. Does the efficacy of the vaccine itself change with age? In other words, in old animals, is the vaccine less effective? And then does rapamycin impact that? Okay, so first thing I want you to look at is the black bar, young without immunization. Let me see if I have up on the screen. I can just stand here. This one right here. So those are the young mice that never got a flu vaccine. And only thing to appreciate is after the vaccine, 2 weeks after that, they were challenged with a lethal dose of influenza. So those mice died because they were given influenza and they did not get a vaccine. That's what we would expect. Those are the young mice that got the vaccine. What does that tell you? Vaccines work. Those mice all survived, right? So they got the lethal challenge with influenza, but they'd gotten the vaccine, so they survived. Okay. This line are the old mice that never saw rapamycin. So they're 24 months old, so that's even older, maybe 65-year-old mouse, 70-year-old mouse. They did get the vaccine, okay? So these mice were all vaccinated, and what you can see is 2/3 of them died with exactly the same kinetics as the young mice that didn't get the vaccine. So what that tells you is, at least if you're a mouse, 70-year-old mouse-ish, there's about a 2/3 chance your immune system is not going to respond to that vaccine, and you will die if you get influenza. Okay. Now, it's hard to see, but on the top, there's a dotted line here. Old plus Rapa. Those are the same aged mice. The only difference is they got 6 weeks of rapamycin before they got the vaccine. And you can see they're right on top of the young. So to me, this is really clear evidence that at least in mice, rapamycin can rejuvenate the decline in the immune system with age, at least as measured by a vaccine challenge. Okay. Wouldn't it have been great during the pandemic if we had something that could do that for elderly people? There's evidence that rapamycin does that in elderly people, and I'll come to that in a minute. Okay, last experiment I'm going to show you. This is also from my lab. We were really just looking at lifespan, and the question was, would transient treatment with rapamycin, like what I've shown you for heart and oral health and immune function, would that do anything to lifespan? So we took 20-month-old mice, we gave them rapamycin or a control for 12 weeks. This was done by Alessandro Bito, who's pictured there. He was a postdoc in the lab at the time. And then we put the mice back on the shelf and let them live out the rest of their natural life. And what you can see, hopefully, is there's a difference between the blue line, which are the mice that did not get rapamycin, and the red line, the mice that did get rapamycin. In this particular group, it was about 60% increase in life expectancy. That's a smaller increase if we're talking about lifespan, but this is remaining life expectancy from the treatment period. So one interesting thing is the treatment period was 90 days, but these mice lived on average 140 days longer. So the increase in lifespan was actually longer than the length of time the mice saw the drug. There's some interesting biology going on there. I have no idea whether this is going to work the same way in dogs or people, or if it does, that it's going to be a linear extrapolation. But if it were, that would be 2 to 3 years for a typical 7-year-old dog, or about 2 decades for a typical 50-year-old woman. So potentially large impacts on life expectancy from a single transient treatment with rapamycin. Okay. So why the heck aren't we all taking rapamycin, right? If it had this effect, wouldn't that be great? There's a lot of reasons. I think the big one is rapamycin has a bad reputation because of how it was developed as an organ transplant drug. If you ask most physicians, they will have no idea of all the data that I just showed you or the human data I'm going to tell you about in a minute. They're going to know about rapamycin, if they know about rapamycin at all, as an immunosuppressant used in organ transplant patients. They'll be like, why the heck would you want to take that? So that's a big challenge. Rapamycin's off patent. So even though there are tons of good clinical trials that should be done, Big Pharma isn't going to spend the money to do the clinical trials to get a label change or label expansion because it's off patent and they can't make a lot of money off of it. This is not so much of an issue, but it is something that is true. There is currently no regulatory pathway at FDA for drugs that target aging. You would have to pick a single indication. If we think this is really slowing aging, it's not clear how you do a clinical trial to get FDA approval for that. And then, as I've already mentioned, if you went to your personal doctor and asked about rapamycin, they're probably going to look at you like you're crazy. They tend to be risk averse and are not used to treating aging as a treatable condition. That's starting to change, but there are all these things, I think, that make it challenging to understand where does rapamycin really work, who does it work for, and even if you want it, getting access to it right now. So what is the current landscape? And that's going to be sort of the rest of my talk, is just trying to give you a feel for where we're at today. So I already talked about this large body of preclinical literature. I think there are at least a dozen obvious endpoints that should be tested in clinical trials for rapamycin in humans today. Like, this should have happened a decade ago, but it didn't. There are some interesting studies in veterinary medicine that are preliminarily looking very promising, and I'll talk just for a minute about the work we're doing in the Dog Aging Project. Most of the human data we've got, though, comes from anecdotal N of 1 studies or collection of information from people using rapamycin, what's called off-label. So this just refers to the fact that in the United States, if a drug is approved by the FDA, physicians can prescribe it off-label for something other than its label indication if they think it's in the best interests of their patient and their patients are fully informed. So there are I don't know how many, 10,000 plus people in the United States taking rapamycin now off-label. And some of them have shared their stories, and we've tried to collect data from them. Obviously, that's not super satisfying, right? We would all like to see the randomized, double-blind, placebo-controlled clinical trials. But I do think we can learn some information from those kinds of studies. And then there have been a handful of small, underpowered, usually phase 1 or phase 1/2 clinical trials, which are nice for evaluating safety, but never really get us to the finish line in terms of understanding efficacy. Okay, so what do we know about rapamycin in companion animals? So I'm part of something called the Dog Aging Project. That's a large observational study of more than 50,000 dogs around the United States and owners. These are companion dogs living with their owners, nothing to do with dogs in the laboratory. So most of that's observational, but we have a single clinical trial called Triad, Test of Rapamycin in Aging Dogs, that is enrolling 580 dogs into a randomized double-blind placebo-controlled veterinary clinical trial. I'll come back to that in just a second. If you're interested, we've published the methods paper. It's a pretty complicated clinical trial, as you can imagine, but I'll tell you a little bit at least about where we're at. And then there's another part of the Dog Aging Project that focuses on cognitive function called the Brain Health Study. This actually was featured in a 60 Minutes episode just recently. I'd encourage you to go check it out. You might recognize this guy. Right there. Stand up, Evan. I'm going to embarrass you. So that's Evan McLean. He is an associate professor here at the University of Arizona. You can see he's hanging with Anderson. So that was pretty cool to get to see some of the cognitive tests that Evan and his team have developed. So anyways, check out the 60 Minutes episode if you get a chance. But part of that study is the woman there, Steph McGrath at Colorado State, is testing whether rapamycin can impact cognitive decline and dementia in companion dogs. Okay. So in dogs, we've completed 2 safety trials of rapamycin. One was 10 weeks, one was 6 months. I can tell you we didn't see any significant adverse events. Really, adverse events were no different from placebo. You're going to see that's a theme that recurs as I'm taking you through some of the studies that have been done. At the appropriate doses, rapamycin seems extremely safe in people and dogs. In our first study, we did see improvement in left ventricular function that very much matched that study from Peter Rabinovich's lab in mice. We also saw owner-reported improvements in activity and quality of life. The owners were blinded. So again, the owners didn't know, the veterinarians didn't know, the dogs presumably didn't know if they were getting rapamycin or placebo at the time the data was collected. Okay, so now this is Triad. So we're doing this study now. About half the dogs are enrolled, 580 dogs, half get placebo, half get rapamycin. So they come in for a pre-screening visit. So there are a fair amount of criteria that are exclusionary here. The dogs have to be at least 7 years old. They have to be between 40 and 110 pounds. They can't have significant pre-existing chronic disease. So this is a study of normal aging, not of kidney disease or dementia. And both the dog and the owners have to be cooperative. You would think that wouldn't maybe be as much of an issue as it is, but that's really important. So assuming the dog and the owner pass the screening part, half get randomized to the placebo, half to rapamycin. This is block randomized by site. We've got something like 20 sites around the United States right now. And then every 6 months and baseline, the dog gets an evaluation. This is a combination of veterinary evaluation, biomarkers, so blood for metabolome, epigenome, microbiome from feces, standard blood chemistry biomarkers. Half the dogs get echocardiograms at each visit, half get neurological exams, and then we have owner surveys that the owners are completing on a monthly basis to track side effects and other owner-reported changes. So that happens for 3 years. The first year, the animals are on treatment. The second 2 years are follow-up. So 3 years, the reason it's 3 years is because we believe that gives us statistical power to detect a 9% change in lifespan. So lifespan's the primary endpoint, but of course, we're interested in a variety of healthspan metrics, disease incidence, so we're tracking secondary and exploratory endpoints as well. And our dose is 0.15 mg per kg per week if you're interested. Doses don't translate well, as you probably know, across species, so that doesn't mean anything for the human dose. Okay. The other thing I want to mention, though, is that as we've been doing this study, a company called Trivium Vet that I was an early scientific advisor for created a veterinary formulation of rapamycin. We are using their veterinary formulation in our study. And they did it from the geroscience sort of perspective. They wanted to evaluate rapamycin because they thought it slowed biological aging. But in order to fit into the FDA framework, they had to pick an endpoint an age-related endpoint that they thought they could show improvements for. And they looked at cardiac function for the reasons that I've already told you about. And so they looked specifically at hypertrophic cardiac diseases, dilated cardiomyopathy in dogs, hypertrophic cardiomyopathy in cats. They started both of those studies with academic centers. They had a problem at the academic center for the dilated cardiomyopathy study in dogs, so that got slowed down. They got early preliminary data in cats, and that's what they ran with. And they got FDA approval last year for rapamycin for hypertrophic cardiomyopathy in cats. So in my mind, this is the first successful clinical trial from a geroscience perspective. So that's pretty exciting, and it shows that that model can work. And I'm not going to get into it, but there are a variety of reasons why the veterinary medicine model is a faster model if you're a company and you want to bring a drug to market than trying to go for human approval. Okay, so I'm just going to wrap up by giving you an overview of the human data sort of as it sits now. And I'm not going to tell you about everything because that would, again, take an hour and a half or more. But I'm going to try to pick and choose what I think are the most informative pieces of information we have. The best randomized clinical trial data we have is not for rapamycin. It's for everolimus, which is a derivative of rapamycin. This is one of those rapalogs I mentioned before. Biochemically, same mechanism. I think it's okay for the purposes of this talk to think of it as rapamycin, but it's not exactly the same drug. So this is work that Joan Manick did when she was first at Novartis and then at a company called Restore Bio. So they did 2 phase 2 clinical trials in healthy older people. So these people were between 65 and 85 years old. And they wanted to ask, could they replicate that mouse study that I showed you? So they randomized the people into a few different groups, different doses of rapamycin. There was a placebo group. And then they gave everyone a flu vaccine. And they asked, were there differences in response to the flu vaccine among the groups? And the answer was yes. The people who got everolimus had a better vaccine response than the people who got the placebo. And they showed that at a once-weekly dose of 5 milligrams once a week, that efficacy was preserved, but the side effects went away. Side effects were no different than placebo at the 5 milligrams once a week. So that's one of the reasons. I'm going to come to like why have we sort of settled on 6 milligrams once a week in people. This is one of the reasons why that once-weekly dosing at 5 milligrams once a week has sort of become the culture, I'm going to say. There's science behind it, but it's certainly not demonstrated to be the best dose by any stretch of the imagination. Unfortunately, the company moved on to a phase 3 trial. Because everolimus had limited patent life, they put a different molecule in the phase 3 that is a different type of mTOR inhibitor, and they did not hit their endpoint. Again, it's a really interesting story. This was November 2019. I was actually at the Gerontological Society of America meeting with Joan when she got the news that halfway through the trial, the board was going to shut down their phase 3 because they weren't hitting their initial endpoints. It was halfway because they were doing the trial in the different hemispheres, the north and south, and the flu seasons are offset. So they had data for half the year. November 2019, you guys all think about where we were in April, May of 2020? Wouldn't it have been really nice to have a drug that boosts vaccine response in the elderly? Missed opportunity, guys. It turns out they went back and did a retrospective analysis, and it looks like even the alternative mTOR inhibitor was working for vaccine response, particularly, ironically, for influenza and coronavirus. So it's interesting. You can check out, there's a paper down at the bottom in Lancet Healthy Longevity that goes through some of that work from Joan. Okay, so that's kind of a sad story. What can we learn from off-label use? So we tried to collect data on off-label use of rapamycin from people who had been getting it from their physicians. I mentioned the most common dose is 6 milligrams once a week. I'm going to go back because this guy. I forgot to mention Alan Green when I showed this slide. So that's Dr. Alan Green. He was the first physician who started prescribing rapamycin widely. Probably, I don't know, 1,000 plus people he prescribed rapamycin to way before it was sexy, right? This was, I don't know, 2018, 2017. Literally, there were, I don't know if Ken was ever you don't have to tell us because that would be a HIPAA violation if you were ever a patient of Alan's. But let's just say there were many people who flew to Little Neck, New York in their private jets to get a prescription from Alan for rapamycin. So he was an interesting dude, but he sort of settled on 6 milligrams once a week based on Joan's studies. And that just became the culture, right? Everybody started doing 6 milligrams once a week. It's varied a little bit since then, but Alan sort of set the stage. He unfortunately passed away last year. Okay, so what can we learn from this off-label use? like I said, 6 milligrams once a week is the most common, but it ranges from 1 milligram once a week up to 20 milligrams once a week. So we collected data from 333 people, all using rapamycin off-label, and we compared it to about 150 or so people who'd never used rapamycin. So we tried to put in a control group. This is an imperfect study. I'm not going to pretend it's anything more than what it is. But I think we can learn some interesting stuff. So one of the things we found was the only statistically significant side effect that was different between the users and the non-users was mouth sores. About 15% of people got mouth sores. That is the most common side effect from rapamycin in organ transplant patients and in people using rapamycin off-label. It's very different, though, because in organ transplant patients, these are nasty ulcers sometimes. In most people using rapamycin off-label, these are more like canker sores. But that is a real side effect, and it is, as far as I can tell, the only thing that consistently shows up as being a side effect of off-label use of rapamycin. Like I said, in our data set, about 15%. Other thing that was interesting is that there were 5 other side effects that were different between the two groups, but they were all better in the rapamycin group. And those were things like chronic pain, depression, anxiety. I don't know what that means. The thing about mTOR, because it's so central in biology, I can go on PubMed and find something connecting mTOR to all of those things. I don't know if it's real. I don't know what the mechanism is, but that's what the data showed us. The thing that I do believe is real is there was significant protection against severity of COVID-19 infection and long COVID. So this was nice because the time range that these people were taking rapamycin during was during the pandemic. And many of them got infected with COVID-19 and continued taking rapamycin. No difference in risk of infection, by the way. Exactly the same between the non-users and the users. But if they were taking rapamycin and they kept taking it, there was a statistically significant reduction in moderate and severe outcomes from COVID-19 and in symptoms that are at least consistent with long COVID. This is a pattern that shows up over and over and over again. Viral infections in particular, chronic viral infections, rapamycin seems to be beneficial for. So I absolutely believe that's a real effect. Okay, some weak evidence for improvements in oral health. So John has gone on and collected dental records from people using rapamycin off-label, done some case reports, can show that it seems as though people using rapamycin don't see the decline in bone that you would typically expect to see in that age range, and some people actually can show an increase in periodontal bone levels. So I would call that weakly suggestive. It's not to the standard that anybody is going to really get super excited about. John's working on a clinical trial, randomized double-blind, placebo-controlled clinical trial. Hopefully he'll be able to get a clear answer there. I'd say there's pretty suggestive evidence in ApoE4 carriers, so for dementia. So for those of you who are familiar with ApoE4, that is the largest genetic risk factor for dementia. It's a pretty high risk factor for heart disease. If you have 2 copies, it's about a 10-fold increase in risk of dementia. If you have 1 copy, that's the ApoE4 carriers, about 4 to 5-fold increase. So there have been a couple of studies now, tiny studies, 4 people, 12 people, right? Not huge. Both of them, though, showed that within just a few weeks in ApoE4 carriers, you can see improvements in cerebral blood flow to the brain, which is important for obviously maintaining cognitive function. And then one of the studies by MRI actually showed increases in volume in the hippocampus and caudate nucleus. And there are dozens of studies in mice showing protection against both normal age-related cognitive decline and in mouse models of dementia. So personally, I think this is pretty real, but the scientist in me has to say this is suggestive. We should be doing these clinical trials for dementia and rapamycin. Suggestive evidence for ovarian function. So there was a study that showed that women who were doing in vitro fertilization got more pregnancies if they'd been given a pretreatment with rapamycin. Again, mouse studies showing that you can reverse ovarian decline during aging with rapamycin. And then there's a study out of Columbia that Yushin Su and Zev Williams are running that just completed. So I know what the answer is, but I can't tell you. But I've got a smile on my face. So we'll get some, I think, more data in a randomized clinical trial setting. It's a good clinical trial. Those are in women who had premature ovarian failure. So that looks pretty promising. I'd say there's suggestive evidence for muscle mass. For those of you who know mTOR biology, there's a lot of concern that people taking rapamycin would lose muscle mass because mTOR activation is required for muscle synthesis. Again, at the doses we're talking about and the way it's being given once weekly, I don't think there's any real concern about losing muscle mass. There was a study called the Pearl trial that was done. This is a distributed clinical trial. A company called Ageless RX ran this trial. It was a placebo-controlled, so half the people got placebo, or a third got placebo. 2 doses of rapamycin tested. It was a signal for improved body composition in women. I'm just going to leave it at that. This study had a problem, though, that they used compounded rapamycin that did not have an enteric coating. It turns out rapamycin gets broken down at low pH in the stomach, so that was a mistake. So they don't actually know what the effective dose was in that study. It was lower than what they were shooting for. And then this study I thought was really super interesting, and this fits with the viral model that I mentioned earlier. So this is in chronic fatigue syndrome, and this was all people using rapamycin off-label. So 86 people, so reasonable number of people, 6 mg per week for 3 months. They didn't have any serious adverse events. They did not see any changes in fasting glucose, A1C, or lipids. So again, that's important because in organ transplant patients, that's something that people worry about. I have not seen any evidence that rapamycin off-label doses at a population level perturbs metabolic or lipid biomarkers. Almost 3/4 of the people showed significant improvement in their chronic fatigue syndrome. That's a pretty striking result. What was even more striking is those people were greatly enriched for chronic fatigue syndrome post-viral infection. So I didn't know a lot about chronic fatigue syndrome before I read this study. And then I did some homework, and I learned that many chronic fatigue syndrome cases are post-viral infection. So this gets back to the idea that there's something about the biology of chronic viruses and rapamycin where that's a population where rapamycin seems to be particularly effective. It turns out rapamycin does boost antiviral gene expression. So an easy explanation would be there's persistent expression of these of the viral genes in people who have chronic fatigue syndrome, and rapamycin is impacting that. I also think it's just a general dampening of chronic inflammatory and autoimmune signaling. Okay, so this is my last slide. This is sort of where I'm at, and as you can tell, I think there's a there with rapamycin, but I really would like to see more quality data. So I'm pretty confident that this sort of 3 to 8 mg per week for most people has minimal side effects, not really any real risk of metabolic side effects or infection. Nothing has come out that is statistically significant yet. However, everything's individual. There are certainly people who are likely to see changes in glucose, changes in lipids, be more susceptible to probably bacterial infections if they're taking rapamycin off-label. I think the immune function effects here are, again, sort of a double-edged sword, right? We know with age there's an increase in chronic sterile inflammation, right? Your immune system responding to self-antigens or other stuff it should not be responding to. But we want our immune system to function normally, surveil cancer, deal with pathogens, right? So I think the key here is to find the dosing strategy and the timing strategy where we blunt that chronic sterile inflammation without impairing the normal function of the immune system. And actually, by blunting that sterile chronic inflammation, we restore the loss of immune function that happens with age. At least that's my hypothesis for what's happening. And I think the once-weekly dosing is partway there, at least. So I think that's why we see improvements in chronic fatigue, things like that, protection potentially against things like long COVID, because that dosing schedule is blunting the chronic inflammation and allowing the immune system to function more like a youthful immune system. And that's why I think there really is pretty good evidence for some autoimmune disorders and things like long COVID, chronic fatigue, that some people are going to benefit from rapamycin. I'm excited about the brain effects. I definitely think there's an opportunity there. I'm excited about the periodontal effects. I'm excited about the ovarian aging. Unfortunately, we just don't have the large data sets that we need to really be confident or to be able to stratify people and understand who is this going to work for, who's it not going to work for. Then I'll leave you with muscle. And again, this is my speculation. Again, I know about data that I can't tell you about yet. I don't think there's any negative impact of off-label doses of rapamycin on muscle function over the long run. I don't know if it's going to improve muscle function. It might in elderly people, or muscle mass. I don't think there's any negative effect. I do think that if you're taking 6 milligrams of rapamycin once a week and you have been sedentary and you suddenly start exercising and you start eating a healthier diet, you might attenuate those newbie gains in muscle, but eventually you're going to reach the same level. That's speculation, because nobody has done that clinical trial yet, but that's kind of what I think might happen. Okay, so I'm just going to leave you with, why do we need this trial? Thank you, Ken, so much for doing this. Because nobody's stepping up and doing it. There's so much evidence, in my view, supporting the need for these kinds of clinical trials. And I think this clinical trial has the opportunity to really fill a critical gap. So I'm going to shut up because I went on longer than I was supposed to. I'm going to let Bonnie tell you a little bit about the clinical trial. And then if we have time, I'm happy to stick around and answer questions.
Thank you very much, Matt. Before we go on, though, before I talk, and I'm going to try to make mine a little shorter, we're going to talk a little bit about the trial, but we have a little gift. All right, so I'm just going to talk briefly about the clinical trial. Very excited. It's building on a lot of the work that Matt just told us about, and we're trying to be as encompassing as possible. It's pretty big, so we're doing a randomized, double-blind, placebo-controlled trial. We're giving weekly low-dose rapamycin or a manufactured placebo, which is being manufactured now. I just got the tooling specs, and we're actually starting to pop out those first placebo pills in the next month. So I'm really excited about that. We're actually going to have people take rapamycin for 2 years every week, and then we're going to have 1 more year of follow-up. We're doing follow-up in the first year pretty frequently at months 1, 3, and 6, and then every 6 months after that. Through the 6 year or the 3 year period. It's 720 people, equal dose to placebo to control, or placebo to drug. And our primary endpoint is the transition from a pre-frail to a frail state, which is like, from a person standpoint, this is extremely important for older adults. It is that transition point that could be the difference between living alone and not living alone, having good functional status or not having good functional status. So it was amazing to learn all about, like, from the geroscience community, the clinicians, and also patients, that this is one of the most important outcomes for them. So that's one of the reasons that we're using that as our primary endpoint. We're actually measuring that frailty transition using the Fried Frailty Index, which is, I should have known that this doesn't work because Matt already showed it. So those are the 5 scoring metrics for frailty. And then we're also balancing that by the immune profile. And this is still to be determined, but we have some very, very smart researchers who are working on developing a profile for us. But what we're trying to do is make sure that we're balancing by people who have like a robust immune system, those that might have a hyper-inflammatory immune system, and possibly even an immunosenescent immune profile where they're starting to sort of see that lowering of their immune relationship. And we're also balancing on comorbidities and sex and age deciles. Very exciting. So our primary endpoint is a co-primary endpoint with Fried being the first, but we're also measuring IL-6 levels. Both the frailty and IL-6 serum levels are really highly correlated. And IL-6 plasma, having elevated levels is indicative of a chronic inflammatory state, and these could be associated with accelerated brain aging, metabolic disease, disrupted metabolic health, and cardiovascular events. So these two things taken together are a really nice mix. We're capturing a lot of things, but these are our primary endpoints. It's really noteworthy. I cannot stress, again, more often how important that transition is for people going from pre-frail to frail. And we have data from the Precision Aging Network study, which is also here at the University of Arizona and 3 other sites around the country. And what we found is we found about a 23% prevalence rate of that pre-frail population, which is pretty consistent with all of the other literature out there. So if we're recruiting from here in Arizona and in Tucson and specifically, we're going to probably see approximately that same prevalence of that pre-frail population. And this is the data that shows the correlation in IL-6 and that pre-frail state using 2 different plasma biomarkers to measure IL-6. And the interesting thing is that both of these are pretty consistent, that as you go up in the robust frail state to a pre-frail state, you start to see an increase in the IL-6 level. Here's some brand new data, hot off the press. One of the things that Matt was talking about is dose. Nobody knows really what the appropriate dose is. So we did a small pharmacologic study that just closed out. And what we were trying to measure is the actual target, the mTOR target, using spectral flow cytometry. Hot off the press as of last week, and even this morning, I was talking to some people, Christine Bradshaw in the Nikolic lab, and she did all the flow cytometry for us. And this is like data that nobody else has ever seen before, so you guys are getting the sneak peek. But I do have to thank all of the people on the screen. There's no above the fold or below the fold here. We have a really awesome clinical operations group and scientists that helped obtain this data. As Matt told you, he did a survey, and the most frequent dosing was 6 milligrams per week, and that was in 42% of the people from his study. And then there's the Pearl study, and actually I have the data that shows what their formulation equivalent was to the true rapamycin dose, and for their 5 milligram, it was about 1.4. So that is way lower than what they had anticipated. And then in the 10 milligram dose, it was 2.89. We are not doing that. We're using the available manufactured drug at 2 milligram dose pills, and then we are manufacturing the placebo on our own. Well, not on our own. Okay, so we had 50 people, 65 years and older, randomized to either a 4 milligram per week dose or an 8 milligram per week dose. We also measured trough levels about 6 or 7 days after they took their dose, because we were trying to make sure it was more of a safety evaluation. We wanted to make sure there was no cumulative effect of rapamycin if you're taking it for a fairly long period of time. We saw no immunosuppressive activity, so that was good. Safety was good. And then we measured 2 immune targets, a phospho-S6 kinase, which is a direct mTOR pathway in the mTOR pathway, and also phospho-AKT. And what we found was on the left side is the S6 kinase, and the red line is the 4 milligram per week dose, and the blue line is the 8 milligram per week dose. And what you can see is that the slope of going from baseline to 8 weeks of dosing, there's a faster rate of change of the actual target, the S6 kinase, phospho-S6 kinase, in the 8 milligram per week dose compared to the 4, which is good. We didn't see any changes on the right-hand side, which is the phospho-AKT. We've seen literature to suggest that that was going to be the case. We weren't really sure, but we're not seeing a difference between those 2 groups. It's okay. Like I said, the literature right now, especially in humans, there's just not a lot of data. But I am very excited because it has really never been shown that we could actually measure the phospho-S6 kinase in a real way in people who are dosing with rapamycin. So this is the first time that anybody's ever seen these data, and I think it's going to be a great paper. So it's noteworthy we didn't see any safety issues. We didn't even see anybody dropping out because of the mouth sores. And granted, it was only an 8-week period of time. We did have 4 dropouts. There were 2 in each dose group. They were not actual adverse events that we would have expected based on any of the literature. We didn't attribute any of those to the actual drug, especially since they were equal in both dose groups. We also genotyped because we were looking for fast and slow metabolizers because, you know, if you're going to give somebody a 4 or an 8 milligram per week dose, if you're a fast metabolizer, then it could actually be more than what you think. And it could have, we were hoping we didn't see any increased side effects in those people that were on 8 milligrams per week. We didn't see that. And then also there's slow metabolizers. So people who might not have measured target hit because even though we were giving them, say, 8 milligram per week, they weren't really getting that much. We didn't see any of that, which is great for us for the trial because it doesn't seem like we need to actually monitor phenotype as we're going through our clinical trial. We had a couple people that we're not really sure. They had mixed results. So in the genotyping, but I'm pretty confident that probably isn't going to be an issue. And then I want to just talk a little bit about our opportunities going forward because we have to have a primary endpoint because this is a randomized controlled trial, and we are actually going to be monitored by the FDA because this is the longest human-based clinical trial that's ever been done. So the FDA is very interested in making sure that we're appropriately monitoring our participants. However, we have a laundry list of things that we are actually measuring because we're really trying to make sure that this is one of the most comprehensive studies that's ever been done. We can't look at everything, but we're really looking at some pretty cool stuff based on, in part, with a lot of conversations from people like Matt and around the University of Arizona. This is just a small group here of all of the people that we've been talking to over the past 2 years, but we've talked to geriatricians and people who work in older adults that helped us define our resiliency measures of what is it both functionally and psychosocial that really matters to older people, right? So I've listed some of those clinicians up at the top of the fold. We also have a Coit Center for Longevity and Neurotherapeutics here at the College of Pharmacy. And so we've got people working on epigenetic aging markers for us. And then we're doing some really cool stuff in the chemical aging, things that you can measure on the skin, which are environmental changes that have been correlated with some of these comorbidities, you know, like diabetes, like cardiovascular disease. And so we're doing autofluorescence of the skin, and we're also doing UV light exposure of the skin to see if there's wrinkling and other kinds of things that you can just measure using UV light. This is the first time that as far as I know, that people have ever done this. So I'm very excited about this idea that we can combine some of these functional and psychosocial measures of resiliency along with immune aging and chemical aging and epigenetic aging and even common blood biomarkers like chemistries and CBC, chem panels, things that we measure all the time. The idea that we can combine all of those to get a profile of normal aging and then see if there's a difference in those people that might have some of these profiles in those people who are likely to respond to rapamycin. And not just rapamycin, because we're looking at rapamycin, but we're also interested in possibly those people who may not have a robust response to rapamycin. Could we combine rapamycin with other geroprotectors, metformin, NAD? There's a lot of them on the horizon that people have been talking about, but this idea that we can also evaluate them and use these biomarkers to possibly direct those people who don't respond to rapamycin alone in the future is going to be something that I feel is really, really going to be valuable for this work. And then I wanted to, how did I go all the way to the beginning again? I wanted to give you I have I came up with a quote, Ken, that this was how I was going to roll this out to you today. And it's not there. For some reason, it looks like I have like multiple slides here. There we go. So this is my grand finale quote that from me, in my personal what drives me is that I would love to see in the future that we're striving for generations of superagers, these people who can live a really long time with very few comorbidities, very healthily. And I feel like this is where the longevity space is going. And when we're talking about longevity or health span, this is kind of like our goal.
That's a good question. I haven't seen any data along those lines. There's definitely, and I'm, oh yeah, so the question was, these are lipophilic drugs, and so could it be the case that people who have more fat mass, for example, would take up more of the drugs into adipose, and that might explain differences in pharmacokinetics or long-term side effects, and it's a good point. I haven't seen any evidence. I would be surprised if there isn't data on that from the organ transplant literature where this has been studied extensively. Maybe Bonnie knows about some of this, but.
Well, it is true that some of those smaller clinical trials that we've been talking about and alluding to, this doesn't work at a, thank you. So it is true that some of the other smaller, underpowered clinical trials actually had as their primary endpoint DEXA. For that reason, not just bone mass, but also sort of like visceral fat. We are trying to actually collect that data as well, but our DEXA scan here at the university is over at the co-laboratory on the south side of campus, and that's a little bit difficult for our participants to be driving to, from here over to there. It's in the works where we're actually going to either do a sub-study, an ancillary study from this study for those reasons. I think that correlating that with lipid as a safety measure will be really valuable, Brian, for those reasons, yeah.
But maybe I misunderstood the question, but there's a couple of things going on here. So I think what Bonnie's talking about is, does rapamycin affect body composition? And that's an interesting question too. And so in mice, at least at certain doses, you can protect against diet-induced obesity with rapamycin, but rapamycin impacts fat metabolism, so it's complicated. What I was going to say though is, and this is interesting, if you look at the pharmacokinetics of rapamycin and you compare the once-weekly dosing, like what's going to be done in the clinical trial here versus organ transplant patients, what you see is there's an initial rapid clearance from the blood because rapamycin gets taken into the membranes of cells because of this activity. And then you see a second phase, which is more the cytochrome P450 clearance. And so it certainly is plausible that could be one factor, adiposity could be one factor that influences individual differences in pharmacokinetics. Yeah.
Well, I was thinking about it being maybe biodirectional because there wasn't, it is not like there's been any studies. I was kind of thinking about it if we had we're not doing any kinds of exclusion based on body composition or BMI, but if we had that information, not just BMI, but also just adipose tissue distribution, we could see whether or not that impacted the response to treatment. So I was kind of thinking about it more.
Yeah, that's true. Yeah.
I don't have to talk as loud. If you're ultimately looking for an FDA approval, right, what do you expect that outcome to be? Is it lifespan, or is it some combination of these different comorbidities? I mean, how do you tie it to outcomes in a randomized controlled trial setting? And how is that, I guess, informing the design of your study?
Well, I don't know that it's informing the design of your study. So first of all, a couple things to say. We can talk about the rapamycin case, and then we can talk more in general. Let's just say you had a different drug you think affected biology of aging. So with rapamycin, it's already FDA approved. So there is not really any incentive, because it's off patent, for people to go for what would be called a label expansion, which would mean you do a clinical trial for a different endpoint and you show efficacy and safety there. But let's just talk about the generic case, because I think that's an interesting question. If you had a drug that you thought slowed aging, what would you do if you wanted to get FDA approval? So if you're in companion animals, in principle, you could go to lifespan as the primary endpoint because they age fast enough. And that, I don't know if you've heard of the company Loyal, but if you haven't, you should look them up. They're a really interesting company. They are pursuing 2 drugs for lifespan extension in dogs with FDA, and they're going through the FDA process. They're actually using a pathway that's called conditional approval, where you really have to only show safety first, and then you have to convince FDA what's called reasonable expectation of efficacy, which basically means it might work. So FDA has said in that case, as long as it's safe, and there's a pretty good chance that it might work, we'll give you 5 years on market. So that would be a path in companion animals. You can't do that in people because people live too long. So I think we're still at the stage where you need to pick an indication. And so, some people gravitate towards the big killers: cancer, heart disease, dementia. Personally, I think that's a really high bar. I like functional endpoints. I like things where you see a decline in function, you can show that you improve the function, you can convince FDA this function is important for quality of life. I really liked the Restore Bio study where they were looking at vaccine response. That's a functional endpoint that declines with age. You can make a pretty good case it's important for mortality and quality of life. And if you can show you can improve that in elderly people, you can get FDA approval for that. So I think there are multiple paths that people are thinking about here.
And just for our study, we're not actually trying to get label expansion at all. The FDA is only interested in monitoring the safety because we are doing the longest trial. I do actually think that last week, as part of Tech Arizona, there was an Alliance for Longevity Initiatives that was up in Scottsdale. And this is a kind of a think tank out of DC, which is trying to make aging as an outcome a more relevant, endpoint. And I think that might actually come to play out over the course of the next 6 years as the study's underway. We'll kind of have to wait and see how that goes. But I feel like for us, we're just trying to do the work because somebody has to do the definitive studies to help elucidate all of the biologic findings that we've had for decades now.
And I'll embarrass one more person. So Dylan, stand up. Dylan Livingston from A4LI. Dylan's the supreme leader of A4LI. He actually started A4LI. And so if anybody's interested in the politics of longevity and activities at both federal and state level, Dylan is your man.
Did I capture that right?
Yeah, thank you for a nice presentation and a good sequence. So people who have been in aging for a while have always been impressed by the effects of caloric restriction. Also people in the cancer field, etc. Right? And this works. And so and I feel the rapamycin pathway is not the caloric restriction pathway, right? It's not a drug that mimics the effects of caloric restriction. Are there is there potential opportunities for synergism?
Great question. So I would say that the most likely explanation is that rapamycin is a partial mimic of caloric restriction. So rapamycin turns down mTOR complex 1 signaling. That is one of the many things that caloric restriction does. So I would say they're overlapping but distinct. And I would say certainly this is something that nobody has really studied carefully, but the impact on the whole biology of our complicated system of turning down mTOR complex 1 under a nutrient depleted condition versus a nutrient replete condition, so lots of food, it's going to have different outcomes. So to answer your question, as far as I know, nobody has carefully tried to look at the combination of rapamycin with caloric restriction in, say a mouse. My prediction is that you would probably not be able to do better than caloric restriction alone, but nobody's done the experiment. The other thing that I want to say, though, is I think from a translational perspective, there are obviously many reasons why caloric restriction is challenging to think about implementing in the general population. And I actually worry about some of the ways that mice and people are different in the way they age. And I think at least that extreme caloric restriction that increases lifespan in mice up to 50% would actually be detrimental in people, because mice don't typically die from frailty or fractures. We know in humans that's one of the concerns of severe caloric restriction. Even if you're eating you get all your micronutrients and stuff, you're going to probably lose lean mass. You're probably going to have lower bone density. We know that's a bigger problem in elderly people. So, but that's a great question.
So one way to commercialize a generic drug is a reformulation that has some significant advantage, like lymphatic delivery. Is any of that going on? And as a corollary to that, if you do improve the bioavailability, then are you maybe going to see some adverse effects?
Yeah, almost certainly you would see you would have a higher likelihood of adverse effects because you're going to get a higher effective dose. Obviously, it's going to depend on, can you get tissue specificity and where are the side effects coming from. So there are multiple groups that are looking at different ways of delivering rapamycin in a targeted way to specific tissues, conjugating it to proteins, things like that. There are also groups looking at combining rapamycin with chemotherapeutic agents to get better anti-cancer effects. So that is happening. I don't know of anybody doing that for longevity. The flavor, a variation on that for longevity, is looking for better versions of rapamycin. So things that either more specifically target mTOR complex 1 or have sort of better brain bioavailability if you're interested in impacts of mTOR signaling in the aging brain. So there are people working on derivatives of rapamycin along those lines.
I think with AI starting and going to be incredibly amazing, I just think a study like this will evolve into a tremendous amount of potential here, and we're just setting a standard for doing that. And if this university can pull this off, if I can pull this off, and we can pull this off, that's great, man. That's helping mankind, and that's just great. I make plenty of money, right? I don't need to make another billion, but if I could, I'd give it to Arizona.
We have one more question. We already have the microphone here. So.
Oh yeah, sure. So just going back to the question of synergy, there are approved PI3K alpha inhibitors now that are actually brain penetrant, and people are looking at those for glioma. And you did mention a dual kinase inhibitor.
Yeah.
Was that a PI3K or AKT?
Very good question. Very insightful. So first of all, actually in glioblastoma, I just saw an abstract come across my radar about a rapamycin conjugate or combination specifically for glioblastoma. I know that's not what you're asking, but that reminded me of that. So in fact, I keep pulling people out of the audience. So in the back of the room, Ben Blue, CEO of Aura Biomedical, has found a better rapamycin. So it's an mTOR inhibitor that increases lifespan to a bigger effect than rapamycin. And it is, in fact, a dual kinase inhibitor that hits both mTOR and PI3K. So yeah, I think there's a lot there. I will say the geroscience field in some ways has dropped the ball by not looking outside of rapamycin. As far as I know, nobody has done extensive tests with any rapalog or dual kinase inhibitor for lifespan. So Ben's group pulled this out of a screen of a library of mTOR inhibitors, but people really haven't looked.
Just one quick follow-up question. I thought it was great that you saw inhibition of phospho S6K. And I also thought it was great that you didn't see inhibition of phospho-AKT, because that was your negative control kind of thing, correct?
And the literature is like I didn't really expect it because like I was talking to Christine
Yeah, nothing
about
should have
this
happened.
today, and there's been half of the literature says that they haven't seen it in mice, and the other half said they had. So we weren't really sure if we were going to see it in or not. And I guess from my perspective, I was happy either way. But I liked how we demonstrated that we actually could get the phosphoS6 kinase, which is like massive win for us because nobody has ever shown that in humans. And then, like from my perspective, the fact that we were able to see that and not see the phosphoS6 or the AKT was really interesting.
Very good. Thank you.
And we're doing some other. We're getting other markers, like other T cell markers in our panel. So by the time the paper is actually published, we're going to have a lot more information that's going to probably elucidate some of this even more. I think it's going to be a really impactful paper.